We are now ready to talk about acid-base balancing, and we'll start this by looking at what it means for the body to be in balance around pH, and where we start to get out of balance, how we start to get out of balance. So we'll start by thinking about what is it, or what is an acid. An acid is an electrolyte that, when dissolved in water, will let go of or release hydrogen ions. So for instance, HCl, which is hydrochloric acid, the same stuff that we put in our batteries and into our stomachs, when we dissolve that in water, we wind up with a hydrogen ion and a chloride ion. Now the chloride ion doesn't have anything to do with acid-base balancing, but the hydrogen ion is all about acids. That is what an acid does, is it produces hydrogen ions and dissolves them in, and has them in when we put them into water. And so when we are measuring pH, the H part of pH, that is all about hydrogen ions. We're looking at what is the number, the count of hydrogen ions per volume of the solution. A base, on the other hand, is a material that will combine with hydrogen. And once they're combined, remove the acidic properties of that hydrogen, so that it is no longer a free hydrogen ion in solution. So for instance, something like sodium hydroxide is a very strong base. When you dissolve that into water, it releases the OH ion. This is also called the hydroxide ion. That's the name for that. When you interact or react hydrogen ions with those hydroxide ions, you do a chemical reaction that produces, let's see if you, what is it? If you were to add them all together, you would have two H's and an oxygen. So that's water. So water has, a molecule of water, because there are no hydrogen ions here, because it's all joined together to make a water molecule, it's not going to affect pH at all. As well, the sodium bicarbonate molecule, when you dissolve that in water, that's baking soda, by the way, when you dissolve that in water, it releases the sodium, so now we have a sodium ion running around, and then this HCO3 minus, so that 3 should actually be a subscript 3. That's the bicarbonate ion. And you can react that with a hydrogen ion, and then we get H2CO3, which you're already familiar with, carbonic acid. So that got really messy there, but there's this reaction, and here is this reaction. These are acid-base reactions, where we react hydrogen, the acid, with some material that will bind that hydrogen, combine with it, and remove it from being out there in the solution. So homeostasis in body fluids requires a pH somewhere slightly basic. If we think about the pH scale, it goes from 0 at one end to 14 at the other end, right here in the middle. Dadgummit. There we go. Shoot, sorry about that. Right here in the middle, it is a pH of 7. That's neutral. Down here, it's acidic. Up here, it is basic. So, a pH scale has a range from 0 to 14. Another word for basic is alkaline. And in the middle, 7 is the neutral zone. So it's slightly basic, just a little bit on the basic side of being neutral. And the requirement for pH to be between 7.35 and 7.45 is very strict. We don't get a lot of play around those numbers. And when we go out of homeostasis, we go one of two ways. We either go into a coma or we have severe convulsions and tetanic contractions of the muscles of the body. So bad, either way you go. So where does all this hydrogen come from, these hydrogen ions? Well, most of them are produced in body fluids as a part of all the metabolic reactions of the body. So there's lots and lots of metabolic reactions that have, as a waste material or as an end product, something that is acidic. So as you've already know, you dissolve CO2 into water. We get that material, carbonic acid, which can then release hydrogen ions and produces a bicarbonate ion. When we do cellular respiration to produce ATP from glucose, and we run out of oxygen to do that with, then we go into anaerobic respiration of the glucose, and that produces a material called lactic acid. And lactic acid is acidic because it drops off hydrogen ions. If we go into fat burning mode in the body, then we produce at the end of that process of burning fats to make ATP, we produce these acidic things called ketone bodies. We go into ketosis, it's called, and diabetics frequently wind up in ketosis, and gives this fruity kind of acetone smell to the breath. When we break down amino acids to get energy from them, we produce sulfuric acid, H2SO4, that can then drop off a hydrogen ion to produce hydrogen ion and the sulfate ion. Is HSO4 minus. And when we break down phosphoproteins, so proteins that have phosphorus in them, and nucleic acids, because they have a lot of phosphorus in them, we produce phosphoric acid, which can also release a hydrogen ion. And then also in the foods that we eat, or as we're busy trying to balance the pH of the food that we eat, we might wind up absorbing hydrogen ions directly from the food too. So lots of places that we can get hydrogen ions from. So what happens when we put strong and weak acid and bases into water? Well, if it's a strong acid or a strong base, then the molecules tend to release most of the hydrogens off of them. Or if it's a base, we neutralize all of the acid neutralizing materials will be released from that strong base. So it's a strong base because when you dissolve it in water, it produces lots of these acid neutralizing molecules, or a strong acid because it produces lots of hydrogen ions. Weak acids and weak bases, on the other hand, they have a harder time letting go of those hydrogen ions or their acid neutralizing ions. So it's all about how much acid each one of them will produce in solution, or how much acid neutralizing molecule each one produces, how much base they release. Okay, so strong acids and strong bases, they make a lot of the thing that is either as the acid or the base. Weak acids and weak bases, they only release a little bit. So because most of the metabolic reactions that are occurring in the body do this thing where they make acids as a byproduct, then it is much more likely for the body to experience a shift toward the acidic end of the pH scale than it is for it to experience a shift toward the alkaline end of the pH scale. So in general, not always, but in general, when we're going to maintain acid-base balance in the body, it usually involves either removing hydrogen ions from the body, or reacting those hydrogen ions with something else so that they are no longer dissolved in solution. And so we've got three ways that we can go about eliminating the acids that are made in the body. We do it with chemical buffer systems, and we'll spend the rest of this lecture talking about chemical buffer systems. And we can also do it through physiological means, where we either breathe out CO2 or we pee away hydrogen ions. So let's look at these chemical buffer systems first. A buffer is a material that when we dissolve it in water can help to stabilize pH by one of two means. They can either release hydrogen ions, so if the solution that they're in becomes very basic, then they'll just drop off hydrogen ions, and then boom, we've buffered the drop or the increase in pH. If, on the other hand, the solution that they are in becomes acidic, well, then they have the capacity to bind up hydrogen ions as well. And they remove them from solution, and in response to that, we can increase the pH of whatever that solution is. So these are molecules that can go both ways. They're neither acids nor bases. They can either absorb acid by reacting with it, or they can create acid by releasing hydrogen ions. So they are the first line of defense for maintaining homeostasis in our body's pH. And the cool thing about them is that they act immediately. As soon as there is any kind of shift, either acidic or basic in the body, then they react by responding to that. Either if it's a basic shift, then they release hydrogen ions. If it's an acid shift, then they bind those hydrogen ions. And so it's instantaneous. And we have three means of doing this chemical buffering. We do it with the carbonic acid bicarbonate buffer system. We do it with another molecule that acts almost exactly like carbonic acid called phosphoric acid. It's the phosphate buffer system. And then proteins as well have the capacity to do this kind of buffering. So let's take a look at these one at a time. The carbonic acid bicarbonate buffer system is one that you know already, because you've learned this equation where H plus with bicarbonate ion produces H2CO3, carbonic acid, which then can fall apart into, which then can fall apart into water and CO2. So we've learned this already. So we're not going to worry about this portion of the equation right now. When we're buffering, we're only moving back and forth between these two portions of the equation. So if, should have had an H plus there, if lots more hydrogen ions appear in the solution, then that helps to drive this equation in this direction. So that the bicarbonate ion that we have in solution will tend to interact with these extra hydrogens that are running around, and they'll produce carbonic acid where the hydrogen is bound to the molecule and isn't falling off. While carbonic acid is an acid because it can release the hydrogen ions, in this case, it winds up hanging on to hydrogen ions. When there's a lot of hydrogen ions running around, we shift the equilibrium in this direction. On the other hand, if something comes along and scoops up all those hydrogen ions, that would mean that we've gone into alkaline conditions, then all the carbonic acid that is hanging out will go the opposite direction. And we'll see it falling apart into bicarbonate ion and then free hydrogen ions as well. So under alkaline conditions, we release the hydrogen from the carbonic acid. Under acidic conditions, we react hydrogen with bicarbonate ion to produce lots of carbonic acid. Okay, so hopefully that makes sense. And what gets a little confusing, I think, for people is that we call this carbonic acid. That's the acid. When we put it into water, it becomes bicarbonate and hydrogen ions because the hydrogen falls off. Water gets in there and interferes with the bond that's holding the hydrogen on and causes it to separate. producing hydrogen ions then. They are the acid that's running around in the solution. That's the carbonic acid bicarbonate buffer system. There's also a buffer system around the phosphate ion. And, I should have said back here, we find this carbonic acid bicarbonate buffer system both in intracellular fluids, in that intracellular compartment, and in the extracellular compartment of the bodies as well. Phosphate buffer system is also found intra and extracellularly, and it's really important in the control of hydrogen inside the cells. So it's very important in controlling the H plus in the cells, and it's also important in urine. That's one way that we can get rid of urine, of hydrogen ions in urine. So under an and here's the reaction that it runs. It's going to look really familiar. We have hydrogen ion. It reacts with something called the phosphate ion, HPO4 2-, and it produces H2PO4. It was 2- to begin with. We added a positive atom to that. Now it's only a single minus. So we go back and forth between these two conditions. So in your mind, just imagine here's where that bicarbonate ion would be. Here's where the carbonic acid would be. So this is phosphoric. Well, it's still a phosphate ion. It's just double ion. It's just a different charge of the phosphate ion. Okay, so this phosphate buffer system, we just go back and forth. Either if it's excess or acidic conditions, excess hydrogen ions, then we can just bind that hydrogen ion here to the phosphate ion. So bind these two together, and boom, hydrogen is taken out of solution and turned into this phosphate ion. If things become alkaline, though, that hydrogen ion that we bound can drop back off again. Now it's a free hydrogen in the body fluids, and it's acting like an acid, and it can help to buffer the alkaline conditions. So we now can get balance back again between the acids and the base that was making us more alkaline. So it's just like the carbonic acid system, just a slightly different ion that we're working with phosphate in it rather than carbon. Then last of all is the protein buffer system. Protein buffer systems are provided because, and this is a super cool thing, if you recall, and you can find this in Chapter 2, so if you're trying to see what the structure of an amino acid is, go back to Chapter 2. An amino acid is a carbon. It's bound to an amino group. It's an N with two H's on one end, and a carboxylic acid on the other end. And that's a C double bonded to an O, and then single bonded to an oxygen that has a hydrogen attached to it. So here we have a hydroxide ion, but when you get this C double bonded to an O and an OH, that's the carboxylic acid group, and this is an amino group. So these two ends are always the same, no matter what amino acid we have. And then recall that there's the variable group that may be attached to it. And R could be just a hydrogen atom, or it could be a really complex, 6-carbon chain or ring. It could be lots of things. It could have sulfur in it and other stuff. So the sky's, well, not quite the limit. There are 20 different R groups that could be attached to an amino acid, each one representative of one of the 20 amino acids that we find in proteins. So we find proteins in the plasma proteins, so the albumins are especially important for buffering, and we also find them intracellularly. So there's lots of proteins dissolved in the fluids inside of a cell, including hemoglobin. So hemoglobin does a lot of this buffering for all the carbon dioxide that we have dissolved inside of a red blood cell. So because a protein is composed of amino acids, that have these functional groups attached to them. That's what we call a functional group, is like a commonly seen cluster of atoms that have a function to them. This carboxylic acid group can drop off this hydrogen ion here. So we can let it go, and that would buffer a solution that's becoming basic. Or if the solution is becoming acidic and there's lots of hydrogen ions around, then that O can then pick up the hydrogen ions, again buffering all the acid. The amino group has the capacity to actually bind yet another hydrogen ion, making it a ionized amino group. And then if things become basic later on, it can drop that hydrogen off. So it's also capable of buffering. And then finally, the R group, depending on what's in the R group, may be able to either drop off hydrogen ions or pick up hydrogen ions, again, buffering the acids that we have in the body. So lots of ways that proteins can be involved in buffering as well. And so we're fortunate that that is the case, because we don't have to keep all those chemicals around. We can also just use the proteins that are already there in the body, which is super cool as well. Okay, I think